Assalamualaikum and hi, this is BSN 3202, Critical Thinking and Problem Solving Assignment of DNA Microarray by Sofia Binti Zulkifli. This assignment is given because my 10 years old sister asked me, "What are you doing, sister? It looks interesting. Can we apply it at home? I really want to try, please." So I'm answered, "This is DNA microarray. Yes, we can try it here if you want, but we must find a few materials. You need to help me collect the materials that have similarities as real apparatus and materials used in laboratory." DNA microarray is nucleic acid hybridization. In this process, two complementary strands of a DNA are joined together by hydrogen bonds to form a double-stranded molecule. This helps researchers to compare and analyze the DNA or RNA molecules of identical sequences. In this experiment, we will use a DNA microarray to compare the differences in gene expression levels between cancer cells and healthy cells. This is the overall method or procedure used. This is the materials that we've been collected at our home. Now we can begin to answer our question, what's the difference between a healthy cell and a cancer cell? We will answer this question by looking at the gene expression or which genes are on and off in cancer cells as compared to healthy cells. We will do this experiment with my sister by measuring the types of mRNA found in both types of cells. The first thing we need to do is find some cells. So this is the cells that we found, which is cancer cells and healthy cells on our patient hand. And this Tupperware will act as microcentrifuge machine. There are three solutions: buffer solution, solvent solution, and washing solution. We have scalpel and light scanner. Vortex machine, we use blender. We use double-side tape as oligo-DT beads. Pipettes, pipette and tips, we use straw. Sample tubes, columns. Microarray and microarray scanner, we use mastermind board game. So this is general steps for DNA microarray. Firstly, collect tissue. Isolate RNA, isolate mRNA, make labeled DNA copy, apply DNA, scan microarray, and analyze data. Firstly, we will collect the tissue. We will collect mRNA from melanoma, a kind of skin cancer cells, and healthy skin cells from the same patient. This way, we can look at differences in gene expression in cells that have same exact genetic blueprint. Next, we will extract RNA. We can extract the RNA from both tissue samples by dissolving them in a mixture of various organic solvents. We can separate the DNA proteins and other cell components, so it will leaving only RNA in the solution. So, it will leave only RNA in the solution. Then, we will mix the tissue samples. We'll mix our tissue samples on the vortex. As the vortex mixes the sample, the tissue will dissolve and the RNA will be released. So we just need to place the tube containing the healthy tissue sample and cancer onto the vortex. Then we need to separate the RNA. How? We use centrifugation. The samples have been placed into the centrifuge. The centrifuge will spin the samples and separate the RNA from the rest of the cell content. Next, we need to transfer the supernatant. After centrifugation, it will produce supernatant and pellet. However, RNA is in supernatant, so we only need supernatant. We need to lower down the micropipette so that it enters the RNA containing top phase. So now we collect the RNA from the cancer sample and healthy sample and transfer it into the clean clip. So the samples contain several different types of RNA. As I explained to my 10 years old sister, RNA will act as a single mother and she has 3 children which are rRNA, tRNA and mRNA. However, we only need the youngest child which named mRNA, messenger RNA. Why? Because mRNA molecules we need but not tRNA or RNA always end in a sequence of adenines known as poly A tail. Only the mRNA reflects gene expression, so we will save the mRNA and get rid of the others. So we will isolate the mRNA. We will wash our RNA samples over columns filled with small bits that will only bind to RNA strands that have a poly A tail. The other molecule will wash away, so the tRNA and rRNA will be waste because it already washed away. However, we need to detach the mRNA from the oligo-DT bits. Now, our columns contain strands of mRNA attached to the poly-T bits by their poly-A tails. So, we will wash a buffer solution over the columns to detach the mRNA from the bits. So, now the mRNA already be detached. Next, we will need to make DNA copy and label with dye. We will make a DNA copy of the RNA. As we do that, we will give the DNA some color. We will add some labeling mix to our 2 RNA samples. We use green labeling mix for the RNA from healthy cells. For cancer cell RNA, we will use a labeling mix containing a red fluorescent dye. So, the labeling mix contains poly-3 primers that will bind to polyatoms on mRNA. Next, it has reverse transcriptase, which is an enzyme that synthesizes a strand complementary to DNA, complementary DNA, which is cDNA. And lastly, we have label nucleotides, which will be incorporated into the new cDNA molecule. The nucleotides in this tube have a green fluorescent molecule attached to them. So, reverse transcription will occur. Reverse transcriptase has assembled the label nucleotides into a cDNA molecule and the mRNA molecule has been degraded. So now we only have cDNA. Good work, our tubes no longer contain RNA but now it contains complementary DNA which is cDNA. The healthy cell cDNA molecules are green and the cancer cell cDNA molecules are red. This is what we will apply to our microarray. So, as we know, 2 complementary DNA strands from different sources can pair with another through hybridization process. So this is single-stranded DNA. So it will become double-stranded DNA by hybridization process. Single-stranded DNA on microarray. Stuck to our microarray are a little piles of single-stranded DNA molecules. A single spot contains many identical copies of the same gene, and each spot of the microarray represents a different gene. Our computer database lists which gene is contained in each spot. So, we will apply DNA on microarray. Now let's add the single-stranded cDNA we prepared from the healthy and cancer cells to our microarray. Remember that all the cDNA molecules in our tube represent human genes, and for every molecule of cDNA there is a matching spot of single-stranded DNA on the microarray. So we need to pipette the healthy cDNA sample and cancer cDNA sample onto the microarray. However, we also need to wash the unbound DNA. Now we will wash off the extra cDNAs that didn't bind to the slide. We will place the microarray into the pan of washing solution in real laboratory. Lastly, we need to scan using microarray scanner. We will scan the microarray to see where the green label cDNA from healthy cells bound to simplify things. So, after we press on the scan green button, and the light scanner will scan through the microarray, and the results or data will be shown. So after we analyze data, there will be red spot, green spot and yellow spot. Green, which is healthy, cDNA is hybridized to that spot. Red, cancer cDNA is hybridized to that spot. While green and red will become yellow spot, will hybridize to that spot. So the last image is the merged image between all of those spots. So, red spots show genes that produce more RNA in the cancer cells than in healthy cells that are turned up in cancer. So the merged image shows any spot that contains both red and green cDNA shows up as yellow. A yellow spot contains a gene that hybridizes to both green and red cDNA, which means that gene was expressed both in cancer cells and healthy cells. However, the genes that show up as yellow spots probably are not very interesting to us since their activity doesn't change when the cell becomes cancerous. Okay, that's all for our DNA microarray experiment. So, this is the applications of DNA microarray, which are to genotype genomes through single nucleotide polymorphism analysis. Second, to diagnose pathogenic as well as genetic diseases in humans. To study transcriptome and proteome. To observe DNA mutations. To identify microbes in the environment with the help of species-specific probes. To detect gene expression of mRNAs of all particular cells at different times. To measure changes in the level of gene expression. And the last applications of DNA microarray is to study genomic gains and losses. However, in this experiment that I have been explained to my 10 years old sister, it is to measure the changes in the level of gene expression. So that's all from me. Thank you. I hope my 10 years old sister can apply this in her future. Assalamualaikum.